Assalamu alaikum wa rahmatullahi wa barakatuh. Peace be upon you all, and welcome to this new live stream for our evolution course. And today we will be discussing the genetic code. So I have had a few requests from participants in the course to talk about the genetic code and make an explanation, and then also see how this can be, whether this can be explained or not by the theory of evolution. And so we're going to do that today. So we're going to try to make this today quite interactive. And this is, of course, week 8 or episode 8 of our course. And after this, you can actually check out article number 8 on the course website, where you will find that I have put together plenty of the material that we will be discussing here today. So this, after looking into what I want to explain to you, I found that it is going to be quite difficult to contain it into one live stream. So Mark here is saying, Assalamu alaikum. Assalamu alaikum, Mark. And hence we will be doing it probably on 2 streams. Okay, so today we will understand some concepts about what the genetic code is, and it will give us an idea about why it is actually almost ridiculous to consider that this magnificent system can be the product of the evolutionary mechanism. So let me first remind you that it is a pretty good idea that you would actually subscribe to the course website, and you will find the course website actually running down there in the ticker. So make sure to go there to the course page and subscribe to the course website. It will give you access to the article that we are going actually to go through today. Now, let's consider this. Our body is a collection of a huge amount of parts. And look at the human body, for example. So the human body is composed of 30 to 40 trillion cells, trillion eukaryotic cells. The kind of cells that forms animals and plants is called a eukaryotic cell. And a eukaryotic cell is a complex cell. It has compartments inside it, and each compartment is like an organelle of the cell that does a specific function. It has sophisticated and pretty huge set of genetic material that sits inside its nucleus. And apart from the 30 to 40 trillion cells, there is also maybe more than that amount in terms of bacteria, but they are not part of the human creature yet. They are interacting with our body all the time. And our body gives and takes from this bacteria. And each of those 30 to 40 trillion cells is composed of many other parts. So organelles, as we were describing, and those organelles are made of proteins and carbohydrates and lipids. And there is the nucleic acid and the nucleotides that form the genetic material. For example, if you look at the proteins, a typical eukaryotic cell will have something like 42 million protein molecules. And this is only the proteins. So imagine this. You have 30 trillion cells in your body, and each of those cells has tens or hundreds of millions of parts in the form of molecules, complex molecules like proteins. And all of this system is being orchestrated and managed by your body. And a protein is not just a molecule like oxygen or hydrogen or carbon dioxide. It is a complicated molecule. A protein can be joined into protein complexes that actually act as if they are machines. So imagine that you have a system that is composed of 30 to 40 trillion factories, and each factory has like 100 million machines in it. Just imagine this. We are talking about hundreds of millions of trillions of parts that are operating together. So whatever we are going to describe today in terms of what the genetic system is just the system that manufactures the proteins inside the cell. But what makes those cells work in harmony together, what gives the signals for those cells to work properly, and what makes this whole system anyway, deliver its function is something that we humans are quite far from understanding. And whether we understand the genetic system or we don't, does not explain life the way it really operates, okay? So just keep that in the back of your mind as we are explaining something about genetics today. It's a big topic, so I'm going to chop it into two, as I've said. And let me give you this analogy. Why is the genetic system something that is not explained or cannot be explained by evolution? Because the genetic system is a system, and systems are designed things. A system will have different parts, and those parts will be in a specified interaction together, to the extent that if one part is not a proper part to the system will break down. If, for example, you're looking at a system like your car, and you cannot just assemble any kind of parts to make the car work. If you want a proper gearbox, then you need to have the proper gears, not just the proper gears, but the gears that are from the manufacturer of that specific gearbox that fit with the other gears in the gearbox. So when you see a gearbox that is properly operating, it is very unlikely or impossible that this gearbox, which is something that is very fine and very correlated and interconnected together, can come by chance. It comes as a product of a very intentional and purposeful design. So what we are going to see today is an analogy. What is this genetic system that we're talking about? So what genes do is that they encode what we know as proteins. And proteins are molecules that perform specific functions in the cell. And those proteins can be assembled into what is called molecular machines. And those molecular machines can produce other substances that are needed by the cell. So the DNA is this device that stores the information about how to create the proteins. Not only does it store the information about how to create the proteins, but it also stores the information about how to create other machines that are not formed from DNA, like machines that are formed from another genetic material called RNA. And this material can be used also to produce the proteins. And then the proteins and the RNA can be used to produce other materials needed by the cell. So let me give you this analogy. The cell and its genetic system are like a computer. And this is why I would suggest to you to have a close look at what we are going to see, and to think with me if it makes any sense that what we are going to see now can be the product of a random process, which is random mutations, and a blind selector, which is natural selection. So first thing, we are going to split this. So how the genetic system works, the genes which are on the DNA are transcribed. A process called transcription converts them into RNA, which is another nuclear material that has information. This RNA is translated into proteins through a process called translation. The DNA itself, since it carries the information of the cell, needs to be copied all the time, because a new cell needs to take a copy of the DNA of its parent or its mother cell. So you have three very important processes that are related to the transfer of biological information: the copying of the DNA, which is called DNA replication, the making RNA from DNA, because you can make proteins from the RNA, and this is called transcription, and the making of proteins from RNA, which is called translation. So those are the three processes. Now, let me give you this analogy that myself, as an electronics and communications engineer with a major in computer and control, there is an analogy, and this analogy is even acknowledged by many scientists and engineers and molecular biologists, an analogy between a cell and its genetic system and a computer system. So think of your computer. First, on your computer, if your computer has no data at all, no programs and no data, you cannot do anything but with the computer. And to have this data and to have those instructions, you need to store them somewhere. So you need a hard disk. And if you have so much information, you need a set of hard disks. And on those hard disks, if you have so much information, you need to compress this information and pack it so that it fits on this hard disk. So this is the first thing. And this storage medium of the genetic material is what we know as the chromosomes. And this is the first thing that we're going to see today. The second thing is that if you want to move this information to another cell, you need to replicate. So we are going to see how this massive data transfer and this massive data copying of the storage system of the genetics goes from one set of chromosomes to another set of chromosomes so that you can have a new cell. We will also see that the mysterious issue that you have brain and you have a heart and you have a kidney and you have a liver and you have muscles and you have bones, all of them have cells that are very different from each other, but all those cells have exactly the same genetic material. So we are going to look into something called epigenetics, which causes the same genetic material to produce very different cells that have very different functions that contribute to very different organs. We are also going to see how this genetic material in this data center is stored into the cell nucleus, and how the cell nucleus is not just a membrane, it's not just an envelope, but it is actually a system that is like a firewall, that is like a security system that does not allow anything in or anything out. So it protects the genetic material in a very specific designed way. And we are also going to see that this genetic material itself, if it suffers damage, okay, there is mechanisms for it to be autocorrected. We are going to stop here today, and we are going to have 5 more parts of this model described on the next live stream, starting from DNA transcription to which is like reading the data from the hard disk and producing RNA, and then the RNA itself produced from DNA being compiled or executed to run a program, a process called excision, and then we are going to see how this RNA code produces the proteins, which is the required outcome of the program. And then we are going to see how those proteins are folded, like when you render an image on the screen. And then we also will have a glance about, even after this protein is finished, how it is transported to where it is useful. So we have this 10-part model about the genetic system that we are going to understand. So now let's start with the hard drive. And I told you that this session will be like interactive, and I'm gonna do something a little bit different today. I'm gonna show you very interesting videos about each of those steps, and I will encourage you to have a look yourself on those videos later, especially after you have finish this session, and I will also have the comments in the comment section, and in the article that you will find on the website published under week 8. There is a fast question here. So it says, is genetics control, or actually, does genetics control our behavior? So isn't it deterministic? Where is free will? No, genetics do not control our behavior. Genetics just produce proteins. Behaviors is something else. I'll give you an example. For example, let's not talk about human beings. Let's look at a cat, for example. A cat knows that it will run after a mouse. A beaver knows how to build a dam. A bird knows how to fly. And you look at different animals that do not go to school, that have very sophisticated behaviors, very sophisticated language, and this doesn't seem to be encoded in proteins. So anyway, genetics may account for part of our understanding for how the body works, but it's very difficult to explain behavior using genetics. So now let's go to the first part about this hard disk, the chromosomes that store the genetic material. I'm going to start by showing you this very nice video that will explain how the data in our genetics is stored into chromosomes. So here is the first thing that we will see together today. Just give me a second to share my screen. I'm going to just run those videos from YouTube. And after we see them, I'll take your questions, and we go on into the 4 parts or 5 parts of the model that we are going to discuss today.
In this every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is in this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So this was the first thing I wanted to show you to understand the level of sophistication that is there in the storage of the DNA material itself. So those genes are not just roaming around. They are packaged into those chromosomes. And as you see, they are packaged around those other proteins that are called histones. And those histones are actually very complicated structures, multiple molecules. And then the chromosomes are wrapped around them to form this material that can then be the hard disk for the genetic material of the cell. Now, the question is, how can a structure like this be a product of a random process? Does this make sense to any of you? The second part of our model is to have a look even on how this chromosomes, or how this genetic material, now, as we say, stores all the information. So if you need to have a new cell, you need to copy all the information. So DNA replication is a very important thing. We start as just one sperm and one egg, and they meet together, and then you have all those new cells that come until you are a 30 to 40 trillion cell organism, which is a human being. And likewise for every organism. Even a single celled organism like a bacteria needs to divide all the time, otherwise it will perish due to very easy environmental conditions can kill it. So DNA replication as part of the cell division is an essential process for life. And as simple as the word is just cell division or replicating the DNA, the process of DNA replication is an immensely complex process. And since a picture is worth 1,000 words, I'm going to show you a very nice video here about how this process is, and it will help so much to appreciate it. So this one is a top level, a high level view of how this process works. And let's have a look.
Before a cell can divide in two, it must first duplicate all of its DNA so that it can pass an exact copy to each daughter cell. DNA replication is carried out by a molecular machine called the replisome that pulls apart the double helix and makes an exact copy of each strand. The DNA to be copied enters the production line from the left. The blue ring-shaped molecule is the enzyme helicase, which unwinds the double helix into two strands. The leading strand is copied continuously. The lagging strand runs in the opposite direction with a more complicated copying mechanism. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So this
Before a
is
cell can divide.
this is a high level view of how this amazing process takes place. By the way, do you hear the sounds from the video? I expect you do hear the sounds from the video, right? If you're not hearing the sounds from the video, please tell me on the chat. So the first time I saw this, it brought tears to my eyes. It was this animation. This animation is done by a computer model that actually simulates what happens on the molecular level. So this is a faithful animation. It just has much less details than what happens in reality. Did you see this? Did you see this machine, the helicase, the blue one? This machine in a bacteria, which has a small genome, revolves at a speed of 10,000 RPM, 10,000 revolutions per minute. Can you imagine a turbine that is moving at 10,000 revolutions per minute? And this is in bacteria, and I think it moves much faster than this in cells that have much bigger genomes like eukaryotes. I don't remember the number, so don't quote me on it. So here, Big Boy says, the more we learn, the more it seems that we truly cannot give God his right estimate. Exactly. And when you look at this process of the DNA replication and you see this machine, what this machine does is that it unwinds, because the double helix of the DNA is wound on itself. It unwinds it into two strands, and then you see one strand, because DNA, there is one strand that runs 5' to 3', and the other one runs the other way. So one of them can get replicated easily. This is very high level of simplification, and the other one, it takes it apart, and then a piece called Okazaki fragment gets replicated, and then it leaves it, and then it replicates the other one, and then the other one, and then the other one, and then it stitches, another mechanism stitches them together, and then the separations are knocked off, and then the separations are filled, and then the whole thing is glued together through a machine called ligase, another enzyme. And then this whole thing, then, if it's a bacteria, it's a circular DNA, gets stitched again together, and then the two strands get connected together. This is completely unbelievable. And to appreciate it more, I'll show you this on a higher level of detail. So let's look at this next video, and you will see more about the Okazaki strands and the separation. Just focus on the video. I'm not sure if I talk while the video is running, if you will hear me or not, but let's try it. So here we go. Yeah. All right, let's now share the screen and marvel on this amazing process.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules.
Look at the blue thing revolving.
Copying occurs at a localized region called the replication fork.
Down here you have
the Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex.
Look at how the machine works.
DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom.
This one is the straight one.
Step in DNA
the other one has the Okazaki fragments.
The two strands by an enzyme called helicase.
Look at how this works.
The incoming DNA to unravel.
Look at the machines working.
10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5'. distinguished by the direction in which their component nucleotides
Look at this
join
one
up.
doing the Okazaki fragments and
The 3'
stitching them.
DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
Yeah. Now, this is the kind of thing that when you see, there is no, there is no further comment. Okay? There's no further comment to say. And once again, is it possible that somebody in his right mind will look at this system, see all those machines, the one that is separating the two strands, the other one that is, copying straight here, the other one that does this unfolding and does the Okazaki fragment and then folds it again and puts another fragment, and then it is just amazing. And as you heard in the video, this is a simplification of the process, because if everything in the process was put together, you will not understand anything. For example, the whole process of error correction, because while this thing is moving at 10,000 RPM, some nucleotides may fail to be matched properly. There is a complete mechanism that will detect that an error has happened and it will correct the error. I'll add those extra details even in the article. Can this be a process of random mutation? A collection of errors have caused this amazing machinery to occur and to work in this magnificent way. We keep asking the question. So I'll show you this, and it will give you some more detail. It's a little longer, but it's worth it. And I want to show you this while you're with me. You can still do your homework afterwards, but we're going to spend this hour together just looking at how this is marvelous. This provides a further level of simplification, so you can, and it partitions the process into 3 parts. So there is an initiation part, and then there is the copying part, and then there is the termination part. So you can even appreciate the level of complexity that we have just seen on the high level, and still this one is on the high level. So let's also have a look at this one. This is from Oxford, and it is a very nice illustrative video.
DNA replication is the process by which a duplicate copy of the genetic material is made before a cell divides. The steps in replication are highly conserved in all organisms. There are a few details that differ between bacteria and eukaryotes, but we focus on bacterial replication in this animation for simplicity. The process of DNA replication is broadly described as having 3 phases: initiation, elongation, and termination. Let's begin by considering the initiation phase.
Now look at how complicated it is just to initiate the process.
DNA must first be opened at a site termed the origin of replication to allow access by the machinery that will copy the DNA. This opening is regulated by a series of initiation regulatory proteins not shown here. Once open, the DNA helicase, a ring of 6 subunits, is loaded onto one strand of the DNA at each side of the replication bubble.
You remember that machine, the blue one that was, revolving very fast? I just want you to look at how complicated it is that this ring, is completely surrounding the DNA. So you need to think, if it's a ring, how did the DNA come inside? How did it, stitch it from the beginning? No, it just clamps into it, and just this process of clamping is not as simple as somebody might imagine. So let's have a look.
Establishes 2 replication forks that will move away from each other as replication proceeds. The synthesis of DNA is catalyzed by an enzyme called DNA polymerase. However, DNA polymerases cannot start a new strand from scratch. Instead, they can only elongate an existing polymer of DNA or RNA. Consequently, a primer sequence needs to be made from which DNA polymerase can continue its synthesis.
Just think of this. So the other machine that was in the previous animation, where the single strand is coming from one place and then you have a double strand coming from the other side, is a machine that needs a double strand. But now you have this original double strand, and you are separating it into 2 strands, and each one strand of them needs to form a double strand. But the enzyme that will take this single strand and cause it to be a double strand cannot start from a single strand. It needs a double strand. So the design of the system is that a primer will come, a place where it can start from, and this blue thing here is an RNA fragment that comes and attaches in this place so that the enzyme can come and clamp to a double strand and start its work. So this is what I am telling you about specified complexity. The system understands itself. It understands that this polymer, this enzyme that is going to come, needs a double strand. So the single strand of DNA gets a little piece of RNA so that the enzyme will come and find a double strand, something that looks like that, so it can start. It starts from the right place. Can this happen by accident?
Primers are made by an enzyme called primase. Once access to the single strands of the DNA has been obtained, the primase is loaded onto the DNA. The primase synthesizes a small stretch of RNA by copying the sequence of nucleotides on the template strand.
So our primase friend will come before the replicator enzyme comes, and it will grab RNA nucleotides and arranges them for the next stage to come and find it ready and starts its work.
DNA polymerase will later elongate this small RNA stretch. The next steps allow loading of the DNA polymerases onto the DNA to copy the entire chromosome. The first component that is loaded is the sliding clamp, a ring-shaped structure that binds at the 3' end of the newly made RNA primer. To get onto DNA...
Now look at how this clamp will clamp around the DNA strand.
And encircle it. The clamp is loaded by a clamp loader complex. The 5-protein clamp loader complex binds to the sliding clamp, and binding of ATP allows opening of the sliding clamp ring. The opened ring then encircles the template primer junction at the 3' end of the primer. Interaction with the 3' end of the DNA stimulates ATP hydrolysis, and the clamp loader dissociates from the DNA to be replaced by the replicative DNA polymerase.
So look at what happened. It was closed, and this complex came to it, so it opened, and then it clamped, and then it leaves it, so it closes, and then another part of the machine will come and attach to it. And all of this is like swimming in a fluid. Can you imagine even just getting close to imagine if we as humans try to manufacture something like this, what kind of intelligence and ingenuity will it need?
The 3' end of the primer is positioned in the polymerase active site to allow addition of nucleotides to this end. During elongation, the replication bubble gets bigger as the helicases move apart, establishing two replication forks that move in opposite directions.
Now, the helicase in this illustration does not revolve, but actually this is a simplification, and you will see that they are making it this way to simplify. But the actual thing is that this is a big machine that is connected on top of each other, and it will be shown at the end of the video with the revolving.
The sliding clamp and polymerase unit at each replication fork moves along the DNA as the helicase unwinds the double-stranded DNA to expose the single strands. Let's focus on just the right-hand fork for the moment. The process.
Now look at the simplification of these green things, the nucleotides arriving, and then you need to think, how do they arrive? How does this green machine select them in the proper sequence? All of those details are removed because when you consider all the details, this is completely crazy.
Both forks are identical. Polymerases are loaded onto both the top and the bottom DNA strands at the fork. Because polymerases only synthesize DNA in the 5' to 3' direction, the two strands must be copied in opposite directions. The bottom strand in this fork is the leading strand. The polymerase on this strand moves continuously from left to right, synthesizing DNA as it travels. The top strand.
So this one in the bottom, the direction of the DNA arrangement is the same as the direction it can read in. So it moves from 5' to 3' nicely, but the other one, when it moves from 5' to 3', it will have to stop because it will engage with the direction becoming reversed. So it will go one piece and stop, and then go the next one and stop, and go the next one and stop, and then the system will reassemble this. This is again a simplification, and the right thing is what we saw in the previous video.
The lagging strand, of which just a short stretch of DNA is made by the polymerase elongating in the 5' to 3' direction. After synthesizing this short stretch of DNA, the polymerase then dissociates and a new polymerase binds to the fork to elongate the next RNA primer. So priming and elongation are happening repeatedly on the lagging strand as the replication fork moves along the DNA. Synthesis is discontinuous.
The message here is that the machines that are performing the elongation or this copying process, the system is designed with the matter in mind that the two strands are running in opposite directions, and what is the direction that the copying can happen in. And because the direction of the other one causes this discontinuous copying, there is this process of doing fragments, as you will see, and then attaching these fragments and covering the discontinuities and then gluing the discontinuities. So this is a system that exactly knows what it's doing, and it's catering for the way the system is designed, and hence addressing this design in a specific manner to the extent that the design will cause the required effect.
Stretches of DNA that are made on the lagging strand are called Okazaki fragments after their discoverer. During the course of replication, these fragments are stitched together. When the lagging strand polymerase runs up against a previously made Okazaki fragment while in the process of synthesizing DNA, the replicative DNA polymerase 3 is replaced by DNA polymerase 1, which is 5' to 3' exonuclease activity. This exonuclease then degrades the RNA primer in front of it, synthesizing DNA as it goes. Once the RNA primer is removed, the two sections of DNA are joined by DNA ligase. Now the lagging strand has been elongated by one more Okazaki fragment. While this view of the replication fork is easiest to understand.
Now this is how it looks like really. So it's showing it as if they are two and they are moving each completely alone, but the actual thing that it's one machine, it has two helicases, and it has the two green machines, the polymerases, and the fragment, the Okazaki fragment by Okazaki fragment process is essentially this folding and unfolding and detaching and attaching process that we have seen in the previous video.
In reality, the leading and lagging polymerases are coupled and move in concert with the helicase. This coupling requires one DNA strand to be looped around so that the lagging polymerase can synthesize DNA in the 5' to 3' direction while moving with the fork. We explore this coupling in more detail in a separate animation. Much less is known about the details of the termination of DNA replication. Once the entire chromosome is
There is something else that I actually terminated it early, so I'm going to continue sharing it. There is this final step of, it's called termination. It's not fully described here, but it's fun also to see.
Much less is known about the details of the termination of DNA replication. Once the entire chromosome is copied, the two replication forks meet each other and are dismantled. The ends of the new DNA strands are then joined, again by DNA ligase.
Look at this machine that comes and glues the thing together like it did before.
One strand of DNA shown in grey and one newly made strand shown in green. Thus, DNA replication is termed semi-conservative.
Yes, so we have one very nice comment here. So Doth Zame says this looks like a factory setup, and it is. This is what I was just telling you in the introduction. We have 30 trillion or 40 trillion cells in our body. Each one of them has those machines has like 40 million of those machines inside of it. 40 million, 42 million protein molecules. Each is a machine that does something, or participating in building of something. And then somebody comes along and says, this is there by accident. Have you ever seen a factory that comes together by accident? And when you look into the factory, you find this kind of extremely sophisticated machines, a machine that revolves at 10,000 RPM, 10,000 revolutions per minute. Can you imagine a gearbox of a car where the gears are moving at 10,000 revolutions per minute and it is not properly designed? With the very finest detail in mind, what will happen to this gearbox or to this motor or to this pump or to this whatever machine that you think of if it is not properly and specifically designed? Jazz says, and this is only the tipping point, finding truth. DNA replication is still being investigated, and we are learning more information about this process until today. Absolutely. Absolutely. And I'm telling you, this does not have all the detail. This only has the high-level process. There is error correction, and there's much, much more involved apart from this. Let's go to the next thing, which is this amazing thing, because you have all of this DNA, as I'm telling you, this whole DNA thing, like in the human being, the DNA, our chromosomes carry 3 billion base pairs, and the base pair is like each one of those two rungs in front of each other. 3 billion characters, 3 billion letters forming instructions. Can you imagine this helicase thing revolving and taking them apart? But the miracle is each of our cells has the same 3 billion instructions, but the brain cell looks completely different from the liver cell, from the heart cell. So how come this is happening? So how come this is happening? It's completely amazing. And the way it's happening is because there is this epigenetic control. So you have the hard drive, which is the chromosomes that we have seen in the beginning. You have the copying of the storage system. And then you have a system on top of this system, an operating system that says which software runs in which cell. So the brain cell will look like a brain cell, and the liver cell will look like a liver cell, and the heart cell, and the muscle cell, and the bone cell. Each will run a different part of the process, of the software. So this is an amazing thing called epigenetics. It is like the operating system that is on top of the software that says which software runs. And let me show you this. And it's completely amazing to see the epigenetic system in action. So we will see something called histones and something called the acetylation of the histones. And the histones are what the chromosomal material is wrapped around. And if you wrap the chromosomal material very tight, then those machines that you have seen replicating, for example, the DNA, the other machines that will come to read the DNA to produce the proteins, they cannot access the DNA that is tightly wound around the histones. And if you want this operating system, the epigenetic system, wants the DNA to be readable, a process called deacetylation will happen, and the histones will let go of the DNA, so the DNA becomes what's called open DNA, and the machines can come and read it. It's completely amazing, and the more amazing thing is to see it through this computer rendering that I'm going to show you now. So let's look first at the high level one. Just give me a second. This is, sorry, the molecular one. So this is the low level one, the detailed one. And then we are going to go to the a little bit to the high level.
Here.
So let me now show you, share my screen again and show you this in action. This is at the molecular level. It's a supercomputer simulation of how the system works on the molecular level.
The mechanisms of life simulated through supercomputers. We are inside a cell in a special part called the nucleus. Here we see some DNA, which is encoded with genetic information, including genes, the blueprints of cells. The motions of a vast number of water molecules have been calculated as well to accurately simulate the behavior of molecules inside the cell. DNA is like a long string composed of a double-stranded helix, folded into very compact structures in the cell nucleus. The center of each of these structures is composed of a protein called histone. Each center consists of 8 proteins, 2 copies each of 4 types of histones. Researchers have been working hard to understand their roles. This structural unit, formed by DNA wrapped around the histone proteins, is called a nucleosome. By treating multiple atoms as one single particle, we can increase the number of phenomena in our simulation. Human DNA measures about 2 meters in length, but it can fit in a tiny nucleus of only 10 micrometers in diameter by forming a fiber of packed nucleosomes called chromatin. For the genetic information to be read, the DNA has to be unwrapped from the histones. K-Computer has made it possible for the first time ever to calculate the force required at the atomic level to unwrap the DNA. It is also known that changes in histone state can alter DNA fluctuation. Histones have unstructured, flexible regions. These are called histone tails. When the histone tails undergo chemical modifications, such as acetylation, the structure of the nucleosome changes. For instance, we know that acetylation of specific parts of the histone tails loosens the chromatin structure, making the nucleosomes less tightly packed. The DNA has to unwrap from the histone proteins to allow other proteins to read its genetic information. Here, the tumor suppressor protein, called p53, is about to bind to the DNA. When the DNA is unwrapped, p53 can move along the DNA and read its genetic information. Chemical modifications, such as acetylation and differences in chromatin structure, affect the reading of the genetic information. This allows various cells and tissues, such as red blood cells and nerves, to be generated from the same DNA. The study of the mechanisms by which genes are turned on or off without changes in the underlying DNA sequence is called epigenetics. Studies using supercomputers to simulate the motions and shapes of molecules will contribute to the development of new drugs and research on regenerative medicine, leading us into an exciting new era.
Oh yeah, it is for sure leading us, because we have no idea how this thing, and the whole discovery of epigenetics is very recent. The whole thing. So before epigenetics, we almost knew nothing. Now we know that we have only been looking at the tip of the iceberg. I have here some comments. So Gotted says, nothing can go wrong if God doesn't want it to go wrong, and everything can go wrong if the free will is used wrong. Perfect balance. Hope you're good, Ahmed. Hope you're good too. Thank you. Our friend Otangelo Grasso says, of course, evolution cannot explain the origin of genetic code because it is an abiogenesis problem. It is also an evolution problem, because then you will need to account for all the genetic code that has appeared since Luca. So there is plenty of genetic code that happened after abiogenesis that evolution cannot also explain. So it's not only a problem to abiogenesis, it's a problem to abiogenesis and evolution. The problem to abiogenesis is even worse because there was nothing before it, but even to evolution, it's not an easy one. So as you can see, this is just marvelous. You have this whole system that is generating all of those on and off switches, and those on and off switches will either wrap or unwrap the DNA so that it can be read, so that this cell will be a brain cell and this cell will be a muscle cell. Now the question is, how did the epigenetic system itself come to being? Well, the epigenetic system essentially depends on components that are like histones, which are proteins, and the histones, which are proteins, are manufactured through the DNA transcription and translation process. So the epigenetic system, as far as our understanding, is made of components that are created from the genetic system. So the question is, which came first, the epigenetic system or the genetic system? You cannot say the epigenetic system because it needs proteins that are generated by DNA. And you cannot say that the DNA came first, because otherwise the DNA will not function properly without the epigenetic system. What does this mean? It means that this whole thing has to come about by an intentional design that brings it to existence altogether as a system. It cannot evolve because it is what we can say irreducibly complex, or because it is an integrated system where if part of the system is not there, the system does not exist. And let me show you something. We saw acetylation. We can also have a look at something else called methylation, which is a part of something called gene silencing. And it gives us a message. When God, for example, tells us do not do this or that and this, and it's for your own good, we are now starting to understand that some chemical influences from outside of the body, like, for example, drinking alcohol, like smoking, like having very bad habits, can cause genes that were not previously expressed to be expressed. And some other habits can cause genes that were going to be expressed to be not expressed. So somebody asked me about behavior. Think that your behavior is not controlled by your genetics, but your genetics are actually affected by your behavior. If you follow the way of your designer in life, it will affect how your genetic system will react to your behavior, not the other way around. It is a big conversation in biology and sociology called nature versus nurture. So we have a question here. Is this related to the minimum gene concept? Yeah, the minimum gene concept is what I was responding to Otangelo about, concerning whether the genetics, the information in the genetic code is coming from abiogenesis or evolution. And this is something like evolutionists would like to escape from you. When you tell them where did the information come from, they will tell you it's coming from abiogenesis, and evolution is not concerned with it. Well, no, because abiogenesis will account for the minimum genome that was there in the very first functioning cell. But this very first functioning cell had like 300 something genes. Humans have 25,000 genes. So where did the rest of the genes come from? So yeah, let's have a look. And I think we'll have a look at this one, and we will conclude at this part, and I'll have some more questions from you, and we'll continue this exciting thing maybe next time. So let's look at the methylation thing, which is what I was talking about, that your behavior, okay, following the way of our designer can actually affect what happens to us. Actually, it does affect, not just can. Let's look at this thing called gene silencing. So let's say that you inherit a gene that will cause you this gene can cause a disease, for example. And let's see this magnificent thing called gene silencing now.
So here's the gene being transcribed, and next to it you see acetylated histones, nice open chromatin, and now you catch these little methyl cytosines. Methyl groups is appearing, and here comes the protein. It binds and will attract this protein complex along with the enzymes, the deacetylases, and now acetyl groups are gone, and histones become tight, chromatin closes, no transcription factors can come through, and the gene is silent.
So I just want you to think of this at the end of this live stream of ours. You have a gene. For example, this gene, if expressed, will cause you to have, God forbid, cancer, or will cause you to have this kind of deficiency, or will protect you from something. And your behavior causes the methylation to take place, and this gene that was going to cause you harm is silenced, or the other way around. The gene that was going to save you is silenced, because you are not following the instructions of your designer in your life. So you can go through the literature of what smoking and drinking alcohol and doing the things that God has forbidden, how this affects epigenetics and how this affects gene expression, and ponder that this system is built inside us, including that it can cause diseases, for example, but it will cause them, in many cases, when you are not following the instruction manual of the designer of the whole system. So is it behavior or genes? No, genes are not controlling our behaviors. Our behaviors will cause our genes to act differently. Just like our genes, the same set of genes can cause a brain cell to occur, or a muscle cell, or a bone cell. It can also cause the brain cell to catch Alzheimer's, or that you live sane until the day you meet God. I would like to leave you to think of this after we have seen this magnificence of design. So what have we gone through today? We have gone through the hard disk, the chromosomal material being the hard disk, how it is replicated, and how epigenetics controls it. I'll be a little bit greedy. I will show you something because it's so marvelous, which is called damage repair. Because our genes, the genome of each cell might catch somewhere from 10,000 to 1 million damages a day, every day. And God has put into us mechanisms that is continuously fixing us. So you have like 30 to 40 trillion cells, and each one of them has like 10,000 damages in each genome every day. And God is fixing you. He's putting this inside you to fix you. He can fix anything for you. So yes, prayers count. Let's look into this. And those are the kinds of processes that they can work or not work. Those are things acting on the molecular level. You need to, the atomic level, okay, where uncertainty is maximum. So let me show you this. What happens on the molecular level to fix our DNA, which is continuously being attacked and damaged. For some reason, wait for it just a second. Yeah. So this will show we are in an age where suddenly, for example, we're infested with breast cancer, for example. So this is an example of fixing the genetic material, and it is part of a video that talks about cancer, where in the case of cancer, the genetic material is just not fixed. This is what happens. And let's go and see.
In the nucleus of the cells, you can find the DNA. Every day, the DNA of each cell is damaged thousands of times. If only one strand of the DNA is damaged, this can be easily repaired. PARP proteins bind to the damage and attract other proteins that repair the damage.
Now we need to think how the PARP proteins find the damage in the first place. And there is so much that we don't understand. And things like interactomes, and these are fields of study that are completely new, and we're just starting to scratch the surface. We can just say now, this protein will find the damage and it will attract other proteins. Tell me how this happens really on the ground. Plenty of research to come through.
When damaged DNA duplicates before the damage is repaired, a double-strand break can arise. In a healthy cell, This more severe damage can be repaired with the help of homologous recombination. BRCA1 protein binds the loose ends of the DNA and starts to unravel the DNA strands. Using the undamaged DNA strand as a template, the damage is then repaired. Finally, the two strands are separated, leaving both DNA strands error-free.
Now, is this marvelous or what? Do you remember the second session of the course when we talked about mutation and how RNA is miserable in terms of not being able to correct because it doesn't have the double strand thing? DNA has the double strand, and it has, as part of the system, the mechanisms that understand that DNA is made of double strands. You see this magnificent thing that happens when the system finds that there is a damage and it cannot fix the damage because the damage was moved into replication. It looks for, there is another detailed video where there will be a machine that moves and it finds the damage, and it finds the damage has occurred. It stops, it sends a signal to stop cell replication, and then this process happens. So it looks for the other undamaged copy and copies it in this magnificent way, and then you have two copies that are correct. Now, is this a system that has evolved randomly, or can somebody in his right mind accept that this is a system that has evolved randomly? And if you think of who has designed this, can it be anything or anyone but God? The supreme, omniscient, omnipotent, all-powerful, all-knowing, all-merciful Creator who has created us and put this inside of us. There is no other solution except denial.
In patients with hereditary breast cancer, the BRCA1 mechanism does not work. When a double-strand break arises in the cells of these patients, the DNA cannot be repaired. The persisting damage can cause cells to die, but it can also transform a cell into a cancer cell. This cancer cell is the start of a tumor in which all cells have the same defect. In this patient,
so I don't want to scare you about cancer, but we just need to think that all this sophistication that is built by God on a molecular level that we cannot even see can be stopped at any time. These are molecules, and one cell that has this defect out of your 30 or 40 trillion cells, just one cell, okay, can spread and cause a tumor and kill you. So imagine that if you lived your life and never caught a cancer, 30 to 40 trillion cells in your body, each catching 10,000, at least 10,000 damages per day, and God has fixed 40 trillion multiplied by 10,000 multiplied by the days of your life for you through this mechanism. And somebody will come along and tell you it's chance, it's evolution. Raihan Sonnen says, brother, how you so educated the subject even though you are an engineer, because engineers like systems, and I got fascinated with this. And the closest thing to this is electronics and computer engineering. And that's why I'm fascinated with it. And you spend some time with things and go into the details, and it really helps you with faith in Allah subhanahu wa ta'ala, faith in God. And then I realized how ridiculous is this evolutionary paradigm. It's ridiculous. And people who are learning this in academia, in college, and teaching it, if they're atheists, how can you live with yourself to know and learn this on the molecular level and remain not believing in God? How can you live with yourself is the question. Darsh Zain says, wow, yes, it deserves plenty of wows. Yes, it looks for a healthy backup. The system, molecules, these machines are molecules. And if I show you now the final video on this episode, where molecules, you see it on the molecular level, how are those inanimate molecules doing this? What kind of programming? It's unbelievable. How can it detect damage in a gene? How can it know what is healthy gene? Yes, this is what we are starting. It is completely amazing. It needs somebody from the outside to understand that this is damage. But those proteins, the P35 protein, it will scan the DNA. When it finds a damage, it will detect it. And if it can fix it, it will fix it. If it can't fix it, it will recruit other molecules, and they will find the other strand, and they will do this crazy thing. We cannot even start to think that we can build this kind of thing. And then somebody who completely disrespects his intellect will tell you that this is by chance. Is this, and does it make a difference if 99.999 or even 100% of scientists will tell you that this is by chance? If somebody respects his intellect, he will not accept it, even if every single person in the world of the 7 billion human beings who live on the face of the earth tell you that this is by chance, and you respect your intellect, you cannot accept that this is by chance. And let me show you, Darth Tame, how this looks on the molecular level, because you will be speechless when you see it. So let me show you the P53 protein in action, doing this not in the very high-level animated way, but on the molecular level, where even when we are looking, we do not understand what's happening. How come this molecule understands what's happening? So let's have a look at this. Wait for it, it's coming. Just one more second, please. I'm sorry for this. Yeah. Let's look at it on the molecular level now. So here we go. Now look with me and marvel on how this works. It's a little long, but we're going to stop it in the middle when you catch the gist of it.
Every day we are bombarded by environmental agents that damage things like even the byproducts of metabolism, such as free radicals, can damage cells. These damaging agents can make changes to the DNA in our cells. DNA encodes the information needed to make proteins, which are the building blocks of our body.
We're looking at a rendering at the molecular level now. Yeah?
2 meters of DNA organized within the nucleus. It is wound around special structures called histones, much like thread on a spool.
With the histones we talked about
Damaged
before.
DNA can include single-strand breaks, double-strand breaks, changes to the DNA code.
Now remember, these are the things that evolutionists tell you are mutations, okay? And they are causing evolution. So guess what? The genetic system has systems that will go and find those mutations and fix them. So if mutations are so important for life, why is the system built to fix them? Is there no sense to look at the reality and then find a theory that makes sense? Mutations cause disease and cause cancer. They don't cause evolution and progress of life.
DNA kinks and DNA sticking together. It has been estimated that an individual cell can suffer up to 1 million DNA changes per day.
So I told you it's minimum 10,000. And in this video, they're going for somewhere from 10,000 to 1 million. And you have 30 to 40 trillion cells in your body. Each one can have up to 1 million damages a day out of all those types of damages. Imagine that.
A special sentinel protein called P53.
This is the P53 I was talking about.
In response to DNA damage.
It's recruited. We know it's recruited. How is it recruited? How does it know who's bringing it here? Why does the system care about damage anyway? These are just molecules swimming in water.
P53 slides along the DNA until it finds a critical site. It then binds to this site. P53.
Listen to until it finds a critical site. We know that this is what's happening. We've seen it. Why is it happening is something else. Because it is by design this way, because the system is built to fix itself. Otherwise, we would have disintegrated just in one day. 30 or 40 trillion cells, each one incurring 1 million damages. You have like 1 million, 40 million trillion damages in your body in a day. Goodbye. Your goo in a day. You will not evolve into a spaceman or an X-Man, or you will not be moving chairs with your mind. You'll just become goo and die. You'll not evolve. But why don't you die? Because the system is designed to find those mistakes and fix them. This is the complete antithesis of evolution. This stupid and dumb idea that life has progressed because it accumulated errors.
Then sends the message to halt cell division until DNA is repaired, or if damage is too severe, the cell is destroyed. P53 is therefore known as the guardian of the genome.
The guardian of the genome.
If the DNA damage is able to be repaired, a very complex and highly...
Now ask the question, who created P53? Who created this magnificent machine? No, P53 is not the guardian of anything. The designer is the guardian.
Sequence of events follows, which is orchestrated by 3 main molecular machines. This is machine number 1. It is searching for a broken DNA end.
Searching. A set of molecules are searching. Even the expressions of evolutionary biologists and microbiologists are expressions that force them to consider intent.
It has found the broken end.
It found it. Wow.
This molecule.
It recruits the molecule. Molecules are talking together.
Once active, the molecule participates in a series of interactions leading to machine number 2.
Machines. I was telling you, every single cell has 42 million machines. 42 million.
Here it is again, this time activating another molecule.
Look, this is magnification 5 million times.
Assembling into its active form. Machine 2 builds a chain to link to machine 3.
Look at this. Look at how magnificent this is. This is what we saw on the high level in the other short video. It's just doing this and that.
3, which has also built a chain.
This is things that are swimming in water. Inanimate things at the molecular level swimming in water. This is beyond our farthest dreams in nanotechnology.
Forms from 2 identical halves, which come together and bind. BRCA1 then begins the recruitment of further complexes that carry out the actual DNA repair.
Do you see how the machine is getting
After
built?
a long chain of events not shown here, the DNA is finally repaired.
Wow, magic. It's not shown here because we don't understand it. How this thing is happening. We don't dream to understand it yet, and maybe we will never understand how this thing happens, how these things get recruited, how they come together, how the system cares anyway about anything, and why. But the why is obvious for those who have eyes that see. But those who have eyes that do not see and ears that do not hear and minds that have become entrenched in denial will not see. Let me look at your comments. He says, I noticed a white back. Mark Ford says, the more we humans learn, a while back, that the more we humans learn about biology, the more fascinating stories we have to come up with to convince ourselves of randomness, as it makes no sense. It does not make any sense. Absolutely, you are right. And they are just stories. They're not even good stories, by the way. How can it know? Does it have a scanner? Did the gene call tech support? Absolutely. They just tell you, this. Put these videos, evolutionary biologists and microbiologists and molecular biologists put these videos and describe this in their books using terms like it's searching, it's sensing, it's scanning, it's recruiting, it's assembling. It's doing none of this. The designer is doing this. This is a system built by design. He says P53 was hired by Mother Nature. Yeah, through job search on the internet, apparently. And he says, Dr. Zain, if this was designed by humans, all of these strands would have been so tangled up, it would be unsalvageable, and you are absolutely correct. And tell you something, guys, this I have tried really to keep this course very scientific, but when you look at things like this, you understand what I mean, that science explains nothing. Science can show us, but when we are at a certain point in science, the explanation, if it is not God, it has to be a dumb explanation. It has to be a stupid explanation that randomness will create this. So I hope this hasn't been so long for you, and it has opened your eyes to another frontier. Because if you look at this and tears do not come to your eyes and your heart is not, flattered by this magnificent creation, then there is a fix that we need to do to our hearts. If you don't think that God is fixing you 40 million trillion times a day, and how great a designer he is and how merciful he is that he's doing this, yet he programmed each one of us through another process, that death is programmed in us and we'll eventually meet him. And we will have to stand, after having seen this, in front of him. And every atheist will have to stand who believes in evolution and abiogenesis without a designer, to stand a moment of honesty and submit that he was in denial and pay the price. But we will not pay this price because there is no other solution but a designer, a great designer, a merciful designer. And this is just a third of the story. We have gone through the hard drive, the chromosomes, the replication, the operating system, which is the epigenetics, and the damage repair, which is like the RAID systems of hard disks and error correction mechanisms. We still have so much in this. I just mentioned a 10-point model that I've put together for you, and we'll be going through this together in the next live stream. I will also try to do like I did in the first episode, so try to compact this in an episode, but I was so fascinated with it, I wanted to share it with you interactively. I hope you have all enjoyed this. Please do share this to those whom you love, to those whom you would like to show the greatness of our Creator, of God, the one God. And Raihanul Suman says, thank you, brother, for great work. May Allah bless you. Thank you, brother, for being here. And please, everyone who's on this stream, please do share this. And please do subscribe to the Evolution course. Everything that I do is free, so that you can even send me emails prior to the session. I will put them and prepare the answers if you have questions. And do share this and invite your friends and family and the ones whom you love to enjoy this with us and marvel the marvels of our Creator. I hope you've enjoyed this, and sorry if it has been quite long. And